Hello and welcome to lesson 2.3. We're going to be looking at how we test substances to see what macromolecules are present in those substances. Our objectives for today are: 1, you need to know what macromolecule each indicator is testing for, and you need to be able to describe the positive test results for each indicator. We're going to be taking notes on page 33. We're going to be watching the video. So this is what your notes look like. Here, you're going to color in the most prominent positive color, okay? And then down here, you're going to write any positive test result colors down here. All right, let's get started.
Biological molecules, sometimes just known as food tests. We're going to be investigating fruit juice, egg whites, starch, regular cooking oil, and we're also going to use distilled water as a control. The principle behind this is basically to find out if those different samples contain biological molecules such as starch, protein, etc. In order to do that, we're going to need to use DC-PIP, ethanol, Benedict's reagent, biuret, and also iodine. Let's start with the iodine test, which is the test for starch. First, take your iodine solution and add it drop-wise to each of your samples. Notice that the iodine is a kind of orangey colour, and when we add it into our samples, that orangey colour remains.
Alright, so you're going to write orange for
the clear blue-black changing
color of solution being tested.
We continue adding this to all of our samples, and amongst all of them, starch is the only one that shows a positive result. So, to summarise, we use iodine to test for the presence of starch. You add it drop-wise to each of your samples, and it turns blue-black if starch is present.
All right, you're going to pause it here, okay? Look at this information, write it down, okay? Make sure you fill in your information from there.
Next, let's look at the biuret test for the presence of proteins. Take your biuret solution and add it drop-wise to each of the samples.
Again, the color of solution being tested, or the before color, is a light blue.
If we give it some time for the reaction to take place, we can see in the egg white it turns a sort of purplish violet colour, and that suggests that protein is present. So to summarise, use biuret solution to test for the presence of proteins, add it drop-wise to your samples, and it'll turn purple
again,
or
pause here
protein
and finish filling in your information.
The Benedict's test is the test for reducing sugars. Glucose is an example of a reducing sugar. First, you take your Benedict's reagent and you add it drop-wise to each of your samples. It's a good idea to label each of your samples for this test because you're going to need to put them into a water bath and they might get mixed up. For your water bath, there's a number of ways to do it, but the easiest way is to just boil the kettle and fill up a beaker with hot water. Put each of your samples into the water bath and then wait for a while. It does take some time for a reaction to occur, but if we watch this speeded up, you can see that one of our samples is clearly turning a kind of yellowy orange colour, and this indicates the presence of a reducing sugar. When we put them back into the test tube rack, we can see the fruit juice is the only one that's actually reacted to this, suggesting that it contains reducing sugars. So, to summarise, we use Benedict's reagent to test for the presence of reducing sugars. Add several drops to your samples and then put them into a water bath. Depending on how much reducing sugar is present, it'll first turn green, then yellow, then orange, and it could form a brick red precipitate if there is a lot of sugar present. The ethanol emulsion test is the test for the presence of fats. First, you need to measure out 2 centimeters cubed of ethanol and pour that into each of your samples. With each one, you need to shake it vigorously. After you've shaken your samples, add 2 centimeters cubed of water and then observe the changes. Here in our oil sample, you can see this sort of white cloudiness formed. This is what's known as the emulsion. To summarise, we use the ethanol emulsion test for the presence of fats. Add 2 centimeters cubed of ethanol and then shake, followed by 2 centimeters cubed of distilled water. A white emulsion forms if fats are present. We use DCP.
Alright, so we're going to stop the video there. We don't need to know that last one. And then we're going to wrap up. Make sure you know which of these tests for what, and the positive test results. All right, I'm about out of time on this video, so I'm going to talk to you guys later. Hope you have a good rest of your day, and I will see you next time.